Good morning, dear students. I welcome you all for today's class. Let us revise the central dogma of life. DNA can duplicate itself via the replication process, and the information present in the DNA can be transcribed to the RNA by transcription process. And finally, the RNA, the information present on it, can be translated to form the proteins, that is the translation process. So today's class we will be studying the protein biosynthesis today partly, and tomorrow we will continue with the remaining part of this class. So the content for the class will be translation or protein biosynthesis, which has got the following subtopics: requirements of protein biosynthesis initiation, protein biosynthesis elongation and termination, and post-translational modifications. So in today's class, I shall be dealing on the first two subtopics, that is requirement and the initiation of protein biosynthesis. The specific learning objectives of today's class will be, at the end of the teaching learning session, you should be able to define translation process, enumerate all the requirements of protein biosynthesis, illustrate the formation of charged tRNA, and discuss the initiation of protein biosynthesis with the help of a neat labeled diagram. As usual, I have divided the class into two sessions. The first session will have the first three SLOs, and the next session will have the last SLO. Let us first define the translation process. In simple terms, translation is the protein biosynthesis. A more comprehensive definition will be like this: the process of protein biosynthesis using the genetic information present in the mRNA at the ribosomes is called as translation. It is a process by which the message in the genetic code in the mRNA is translated into the amino acid sequence in the proteins. Now, this translation process takes place in the cytosol, that is on the ribosomes present in the cytosol. Now, these ribosomes may be present freely in the cytosol, or they may be attached to the endoplasmic reticulum to form the rough endoplasmic reticulum. The proteins which are synthesized on the ribosomes attached to the rough endoplasmic reticulum are usually exported out of the cell, whereas the proteins which are being synthesized on the ribosomes freely present in the cytosol are meant to be the proteins of the cell itself. Protein biosynthesis is a complex phenomenon requiring a large number of molecules like amino acids, mRNA, energy in the form of ATP and GTP, the ribosomes containing the rRNA molecules, the transfer RNA, the enzyme amino acid tRNA synthetase, and various protein factors. Now we will study each of them a bit more in detail. Coming to amino acids, for the protein biosynthesis, all the 20 standard amino acids are required. So, amino acids are the building blocks of protein, and we know that there are 10 essential amino acids. So, absence of even one single amino acid, that is the essential amino acid in the diet, can decrease the synthesis of the proteins. Next, coming to the messenger RNA or mRNA. So, mRNA, which is synthesized from the DNA during the transcription process, carries the genetic information. That is, it has all the information required for the synthesis of proteins. Recall the structure of mRNA. So, in eukaryotes, mRNA has got a cap at its 5' end, and it has got a tail, that is poly A tail, at its 3' end. And it is synthesized from hnRNA, that is heteronuclear RNA, having both introns and exons. Now, when it gets converted to the mature mRNA, the introns are being removed and it carries only the exons which codes for the protein. Even this mature mRNA having only exons still has got certain untranslated regions both at the 5' end and the 3' end. So we have what is called as 5' UTR, that is untranslated region, and 3' UTR. Now, in case of eukaryotic mRNAs, each of the mRNA codes for only one polypeptide chain. That is, they are monocystonic. Cystron is the smallest unit of genetic information. That is, one cystron one polypeptide chain. Earlier the concept was one gene, one protein, but now the concept is one cistron, one polypeptide chain. And the eukaryotic mRNA are monocistronic. Whereas if we see the mRNA of the prokaryotes, they are polycistronic. What it means over here is that is in the prokaryotes, the mRNA may carry the information for synthesis of more than one polypeptide chain. And in this mRNA, each of this part of the mRNA may have initiator codon AUG and the terminator codon. And after a gap of few nucleotides, maybe a next initiator codon may occur for the next protein. That is, in generally, prokaryotic mRNA are polycistronic, having the information for more than one protein or polypeptide chain synthesis. Now, this translation process requires a large amount of energy, which may be provided by the energy currency of the cell, ATP as well as GTP. So, GTP is an important energy transducer for protein biosynthesis, requiring both for the initiation and elongation and termination process. Now, coming to the place where the protein biosynthesis is taking place, that is the ribosomes. So, this diagram over here shows the ribosome which is attached to the endoplasmic reticulum, giving the rough endoplasmic reticulum. You can see over here that this is the ribosome having both the larger subunit and the smaller subunit. So recall that each of this ribosome may have a larger subunit and a smaller subunit. Again, this diagram I had put it across under the nucleic acid chemistry chapter. So where it was shown the differences between the prokaryotic and eukaryotic ribosomes. So prokaryotic is 70S, whereas eukaryotic it is 80S. So prokaryotic is made up of 50S and 30S, whereas eukaryotic is made up of 60S and 40S, and the corresponding rRNA molecules in each of the subunits has been shown in this diagram, which had been already taught to you in the previous class. During the protein biosynthesis, the ribosomes have three imaginary sites called as the A site, the P site, and the E site. So these are the three imaginary sites. So A site is the position on the ribosomes where the amino acyl tRNA binds, whereas the P site is a position where the peptidyl tRNA binds, and E site is a position where the deacylated, that is the tRNA from which the amino acid has been removed, called as deacylated tRNA, exits from the ribosomes. So these three sites are the imaginary sites, and you will understand the importance of these sites when we go for the elongation process. Now coming to the transfer RNA, which actually carries the amino acid, which acts as the adapter molecule. So we know that the proteins are in the language of amino acids, whereas the genetic information is in the language of nucleotides. So hence this transfer RNA is the one which is acting as an adapter molecule or the dictionary between these two. Recall the structure of tRNA. It is a chloroleaf structure having four major arms. That is the amino acid attachment arm, the anticodon arm, which is complementary to the codon present in the mRNA. It also has two other important arms, that is the T-CC arm and the D arm. So each of this arm has an important functions in the synthesis of the proteins. You will see as we proceed further. So here the amino acid is attached to the site at the CCA by forming an ester bond with the adenosine of the CCA. Now, apart from this mRNA, tRNA, rRNA, and the amino acids and the energy, we also require a number of protein factors which are important for all the three phases of the protein biosynthesis. That is, we have what is called as initiation factors, so which are abbreviated as IF. So we have in case of prokaryotes three initiation factors, IF1, IF2, and IF3. Whereas in case of eukaryotes, there are at least 10 initiation factors so far discovered, and many of these initiation factors are multi-subunit proteins, that is having a subunit from anywhere from 3 to 8 subunits, and a prefix E is given. E here stands for eukaryotic. So all the eukaryotic initiation factors are written as E, EIF, EIF2, 3, 4, like that, so on. Okay, so there are at least 10 eukaryotic initiation factors, whereas in case of prokaryotes, there are only 3, which are written as IF1, 2, and 3 respectively. Now, apart from the initiation factors, we also require elongation factors. So, in case of prokaryotes, we have EFTU, EFTS, and EFG. Correspondingly, in eukaryotes, we have only 2 of them. EF1A, some literature may give it as EF1 alpha also, and then EF2. Whereas, again, coming to the termination factors or releasing factors. So RF stands for releasing factors. For the termination of protein biosynthesis, these factors help. And in case of prokaryotes, we have RF1, RF2, and RF3. Whereas in eukaryotes, there is something called as eukaryotic releasing factor. So far, only one has been shown. So ERF, E small e, RF stands for eukaryotic releasing factors. So these are the various protein factors which are required for all the 3 steps of the protein biosynthesis. So based on the step, we have the initiation, elongation, and the termination or the releasing factors. So on top, what I have given is the prokaryotes. The second line here is for the eukaryotes. Now, before we study the actual translation process, it is important for us also to study another important reaction, that is the activation of amino acid. So, activation of amino acid, also called as charging of the tRNA, is a process by which the amino acid is attached to the acceptor arm of the tRNA. And during this process, the amino acids are also activated. That is, this is an energy requiring process requiring equivalent of 2 ATPs. It is requiring equivalent of 2 ATPs. That is, during this process, you will see that the ATP gets hydrolyzed to AMP, and whenever ATP is hydrolyzed to AMP, it means the requirement is equivalent to 2 ATPs. And the process also requires magnesium ions. Now, the key enzyme for this process is the amino acyl tRNA synthetase. That is, this is the fidelity enzyme. What it means is it recognizes the correct tRNA and recognizes the correct amino acid for it to be added onto the tRNA. So this occurs with the help of the DHU arm or the D arm of the tRNA molecule. Now say, let us take an example of a tRNA having a triple A at its anticodon arm. Now this triple A can form complementary base pair with triple U of the codon in the mRNA. Now triple U codes for phenylalanine. Now this particular enzyme, amino acid tRNA, recognizes the tRNA with the triple A at its anticodon arm. It recognizes the phenylalanine and attaches the phenylalanine to the tRNA molecule. Hence, this enzyme is considered as proofreading mechanism of translation. That is, the error rate which can occur because of the presence of this enzyme, amino acid tRNA synthetase, is very, very low. So, it recognizes the correct anticodon arm and recognizes the correct amino acid. Now, let us study the actual reactions where this activation of amino acid is taking place. Now it is a 2-step process. That is, the activation of amino acid or charging of tRNA occurs by 2 steps. In the first step, the amino acid here gets activated by hydrolysis of this ATP, and a complex of enzyme, amino acid, and AMP is formed. So the enzyme here is amino acid tRNA synthetase. Now in the second part of the reaction, that is the second step, now this activated amino acid is attached to the acceptor arm of the tRNA molecule over here, and that is to the adenosine of the ACC. Actually, it should be read as CCA, but since it is given from 3' here, I'm telling it as ACC. So that is, it attaches to this adenosine of this, and during this process, the AMP gets released as well as the enzyme gets released. So at the end of this reaction, there is consumption of energy in the form of equivalent of 2 ATPs, and there is activation of amino acid which also occurs by attaching it to the tRNA molecules. So with this, we have come to the end of this particular session. So we will continue in the next session with the initiation step of the protein biosynthesis. Thank you.